All right, so welcome everybody. We're sorry a bit, well, slightly late starting, just had some technical difficulties. And welcome to our April 2025 Developmental Disability Clinical and Research Rounds. And this month, our focus is on research. And we have as our guest speaker, we have the esteemed Professor Dr. Julio Martinez Trujillo. I think and Julio's topic will be today, he's going to talk about developmental trajectories of stem cells, derived neuronal networks, and the applications and studies of neurodevelopmental disorders. And before I introduce Julio, I think probably the best thing I can say about Julio is truly one of the nicest people I've ever met. A couple of years ago, there was an issue with Julio's cross-program in psychiatry, and I wasn't sure if Julio actually had the ability to get mad. And I don't just remember Julio, but I actually emailed and said, look, can you at least pretend to be angry about this? Because it's the only thing to get attention from people. And I've never heard anybody say anything less than Julio's an incredibly nice person. So you're a good human being, Julio. So Dr. Martinez Trujillo is a distinguished neuroscientist specializing in the neural mechanisms of cognition and behavior. Julio was born and raised in Cuba, and went to medical school at the University of Havana and received advanced clinical training in neurology and clinical neurophysiology at the Cuban Neuroscience Center. Julio then moved to Germany, where he received a master's degree and a PhD in neurobiology from the University of Tübingen. And I think that's where Suzanne Schmidt was also, if I'm not mistaken, in part of her studies. And then in 2000, Julio moved to Canada for postdoc at York University Center for Vision Research. And then a few years later, moved to McGill, where he was in Montreal, where he's an assistant professor in the Department of Physiology, and also was appointed to a Canada Research Chair in neuroscience. Julio has been in London since 2014, which is wonderful for us here, and he's now a full professor in the Department of Physiology, Pharmacology, and Psychiatry. He is a scientist at the Robarts Research Institute and holds the provincial endowed academic chair in autism from the provincial government. So Julio's research really focuses on pretty hardcore basic neuroscience stuff, but the neural underpinnings of attention and working memory and social behavior, particularly in the context of autism. His more recent work has extended to the study of stem cell-derived brain circuits in the context of neurodevelopmental disorders. And Julio's academic career has really been impressive, sort of as like the CV we would all like, in sort of top journals, including Nature, Nature's Neuroscience, Neuron, and PNAS. But also beyond his research, Dr. Martinez Trujillo is committed to mentorship and education and contributing to and fostering the next generations of neuroscience and neuroscientists. And his dedication to advancing the understanding of the brain continues to influence both academic and clinical approaches to neurological and psychiatric disorders. So we are very fortunate and pleased to have Julio as our guest speaker today. Dr. Martinez Trujillo, the floor is yours.
Yeah. Well, thank you very much. Can you hear me well? Can you hear me well?
Yes. Yeah, we can hear you.
Hey, perfect. Thank you very much, Rob, for the introduction, for saying those nice things about me. I think I'm probably simply reactive when I interact with you, because I think that you really care about the children that you see and the things like that. And it's very hard for me to be angry at someone like you. I don't know about other people, but that's is something that I say. And to be in this field,
I think other people find it quite easy, I think, actually.
All right. So, okay, let me get into this. So thank you so much for that. Several years ago, maybe 2 or 3, 4 years ago, I got into this field of developmental study of developmental disorder using stem cells, and I did a sabbatical. And I became very interested in this field, and I hope that I'm going to motivate you today and see why this was so interesting to me. So the objective, I just want to go over them, is to understand how adult mature cells can be converted into pluripotent stem cells, and then into a particular cell lineage, like, for example, into neurons. The second thing, to understand that the differentiated neurons obtained from these induced pluripotent stem cells of a certain patient, if the patient carries a mutation, the cells also have the mutation. So, and they're tools to induce mutations in normotypical cells. The other thing is that to know how the neural networks carrying SHANK2 mutations, and we're going to see a little bit more about SHANK2, have developmental trajectories that are different from normotypical networks. And finally, to know that the network phenotypes produced by mutations in SHANK2 can be rescued using pharmacology and probably other interventions, which opens a lot of venues for the future. So first of all, I'm just going to start very basic. Many of you know exactly how embryonic development happens. So you start from one cell that is fertilized, and then you have cleavage, and you have different cell stages of cell differentiation. And here you have, for example, here a blastocyst.
So Julio, I don't think you've advanced your slides.
No?
Nope, still the first one is showing.
Oh, okay. There is something wrong here. Let me just try to do it again and see if I let me stop sharing, and I'm going to share it again, and see if that works. Is it advancing now?
Nope.
No, it doesn't seem to.
Unless you're just playing the same slide over and over.
No. Not advancing.
Nope.
So that's really let me see what else I can do here. Let me just stop share again. Share, and I will say share sound, optimize for video sharing, and I will say share, and now still.
Can you try advancing it?
Is it working?
Nope.
No, it's just stuck.
Let me just see what I can do here. If I would say, this is my window, this is my screen, this is my Zoom, so I say share, and that one, I will share that window, play. Is it working now?
No, still not switching.
Not switching still. That
Maybe instead of presentation mode, you could just leave it up the way it was.
Let me see. Okay, let me just I still have to share. Like here.
Yeah, just like this.
So okay, so I'll try then to see how I don't like this thing here, but I don't think I have much of a chance to hide these tools. All right, so you see now on my screen, and probably there isn't a question and answer something. Are you connected to a second screen? Sometimes that messes up. No, I'm not connected to a second screen. So let me see. Let me just try one thing more. I'm just gonna quit my presentation, my software, and I'm going to initialize the software again. And maybe it's my keynote that has a problem. I will share a screen again. If that doesn't work, I go into presentation mode.
This is why Apple is actually not the way to go.
Never had this problem though, but
it might have something to do with the tariffs also.
Let's see if now, is it working now?
Yes.
Okay, I went over the objective already. Perfect. So it was probably the software. And I went, so I was trying to go very quickly over this. This is the different stages of embryonic development. And the point that I want to make here is that when you have that there are different stages of pluripotentiality in these cells. So the cells could be totipotent or could be pluripotent. A totipotent cell is a cell that can differentiate also into a placenta, component of the placenta or the embryo. A pluripotent cell is a cell that differentiates only into the components of the embryo, basically. So here it is an example, for example, of a blastocyst. So and in this blastocyst contain type of cells that are pluripotent. So basically, they can become part of the embryo. And the type of cells that we're interested in here, of course, they're ectodermic cells. And these ectodermic cells, they are the neural plate originates from the ectodermic cells. And here I don't want to give an embryology lesson here, but you have the neural plate, and here you have the separation between the neural tube, for example, the neural crest and the epidermis, all to the same fate. But the whole idea is that these cells are stem cells, that they are pluripotent, so they can convert into different fates. Now, these cells have the capacity of cells dividing and to develop into the 3 primary germ cells in the adult body. Now, imagine that you have a set of cells that, for example, pluripotent stem cells in the blastocyst, that they convert into different phases. Here you have mesodermic, for example, ectodermic. Here you have the immune system, brain, heart, immune system. And one of the things that is common to all these cells is that they have the same DNA. Many of these cells, they preserve the DNA. And there is a very simple idea that some people look at several years ago, that if they actually have the same DNA, could they be induced or reversed back to a state of pluripotency? So if this is the same genetic program in the cells, the same DNA, so can you do that? And when people figured that out, that this is actually possible, in 2006, Takahashi and Yamanaka opened a completely new venue in stem cell research. So by showing that the expression of only 4 transcription factors, and these are the 4 transcription factors, you don't have to learn them, and happens that only with 3 you can actually differentiate the cells, was sufficient to convert fibroblasts, the skin cells. So they basically have a fibroblast cluster from the mouse, and they convert it into something that looks like an embryonic stem cell, right? And they were named induced pluripotent stem cells. So this is, for example, this is the first paper that they published that was in the mouse. And then this is Shinya Yamanaka. And this is the second paper where they show also that they could do that in humans. So the whole idea is almost like you can actually convert many of these cells, as long as the cell has the DNA intact, into a pluripotent stem cell. And from pluripotent stem cells, you can develop, you can convert it into other fates. It's called differentiation. So they have different differentiation protocols for that. Now, you may ask, well, if we have animal models, what is the rationale behind using human-induced pluripotent stem cells as models to study the nervous system function during health and disease? And this is a good question. So, and I will try to provide a good answer to this. For example, this is just a neural plate, an illustration of how the cortical plate develops in a mouse and in a human. So you have here the neural tube, and there is progenitor cells, the radial glial cells, that they start growing vertically and they start migrating toward what is going to be the cortex. And they have these, they proliferate along here, and there is these intermediate progenitor cells that they are, for example, this is a ventricular zone, the subventricular zone, the intermediate zone, and the cortical plate, and they keep migrating like that, and they here convert into neurons, and how do we get the neurons in our cortex. Now, in humans, this process is a little bit different. In humans and other primates, what happened is that there is this outer subventricular zone where there is an expansion of these progenitor cells. And an expansion of these progenitors may almost look like a fan, like one of these Spanish fans. And my grandmother used to to you because where I grew up in Cuba was very hot. So, and that produces a very large number of cells here. So there is something about humans that increase the amount of cells that actually they develop in the cortex. So, and you might think about genes, so certain genes, so this is what people have found, or certain programs that just make this process different, and therefore it's different. So it's different in humans and in animal models. So this is a good reason to study human cells. Here is another example. For example, nervous systems could be found in many type of animals. Exactly here in the chordata, with the appearance of the neural tube, and in a species like in mammalian species like rodents and primates, for example, you had the development of the neocortex. And two of the largest inventions that they have happened, or development that's happened in the that has been very meaningful for mammal, is the appearance of the CSF and the appearance of this neural tube, where those cells actually can proliferate, like I showed you in the previous slide, and they can form 6 different layers of the cortex. So, but as you can see, the mouse has a much smaller cortex than the humans, than the primate, than the human in this specific case. So humans are special somehow, and they're special because the cells are also special in a way. So, and it's not only amongst the difference between primates and non-primates and rodents. It's also the difference amongst primates. This is an example of, for example, of the different of the genus Homo. That here we have the Neanderthal. We don't have data on that, but we have data in the Homo sapiens, and we have data in other species of apes like the chimpanzee. And if you look at a neuron in the human and a neuron in the chimpanzee, humans, cortical neurons, they have many more branches and many more synapses than the neurons in the chimpanzee. So even with something as close to us like a chimpanzee, neurons are different. And one of the reasons that people think that may happen is due to a phenomenon called bradychrony. The bradychrony is just basically slow development, in which the immature neurons, they convert into mature neurons, but they convert it to a very slow process. And we know it doesn't matter how hard you try to teach your 4-month-old to walk, it's probably not going to walk, or to talk, it's probably not going to talk. So the human brain needs this period of a slow development to produce maybe these large neural trees and these large neurons. Here is a summary of what I have said. There are changes in developmental time in humans. For example, the human neuroepithelium expand due to a slower transition and establish a larger pool of founder cells before neurogenesis begin, before the neurons actually start forming. So therefore, we have more neurons than pretty much any other species. Human neurons show also large dendritic growth and delay acquisition of mature neuronal activity. So they just go slow. If you if you make a culture of neurons, human neurons, and a culture of mouse neurons, the mouse neurons are firing and becoming a network much faster than the human. So, and this is really important. So, and I introduced this definition of bradychrony, which is a slower developmental pace. Now, this slower developmental pace helps human to incorporate variables, environmental variables, into the shaping of the neural types, of the neural development. And as many of you know, the human brain is developing until you are 25, 30 years old. And still the prefrontal cortex keeps developing that way. So, and all of this is to tell you that there is a need for model systems that replicate the genetic architecture of the human brain. So there is really a need for these systems. And one of these systems is, of course, the iPSC cells, the human pluripotent stem cells. Here we have the process is basically you take fibroblasts for these skin cells, and now you revert the cells to a state of pluripotency. That will be induced pluripotent stem cells, and they actively develop. And if you use the right protocols, you can go into different cell types. You can differentiate them into different cell types. And these systems enable the study of genetic programs, enable the study of the effects of mutations and different systems in different ways. These are, for example, another system that you could develop, which is called a brain organoid. And these brain organoids could be developed from different tissues, hippocampus, forebrain, choroidal plexus. There is different markers that are expressed by neurons in these different areas. And you can take these pluripotent stem cells, convert it into neurons, and drag it into one of these fates. So this is where the state of the art of things at this point now. People are developing these kind of things. Now, over the last years has been really a revolution in biomedical research. And this revolution has allowed the development of, for example, 2D networks. And these 2D networks is, you can take stem cells and you can develop into neurons in the presence of astrocytes usually. And then you can develop into a monolayer and you can study how they connect. And that's most of the data I'm going to show. Or you can develop 3D organoids. For 3D organoids, it's kind of funny. The process is to put it in an orbital shaker and put in a matrigel, which is some sort of glue, a polymer that you use for that, for the cells to aggregate on it. And then you shake the cells and the cells acquire this three-dimensional structure. And then they actually could even get together. Now, the whole idea of using these model systems is that now you can have these systems that they contain, for example, very specific mutations linked to disease. And now you can do all the omics, genomics, transcriptomics, and to look at how different mutations affect the development of the single neurons or network of the networks in this case. And you could even take that a little bit farther, and you can try to look at pharmacology, for example, genetic therapies or something like that, trying to rescue these systems, the phenotypes that you see in these systems after some functional measurements. This is also a single cell exploration to look at properties of the nerve. Now, there is something that is also important that I don't want to miss, is that there are at least these four, and there is a fifth factor that I will show that allows us to do the kind of studies that that we do in the lab, and which is the development of genome sequencing, cell reprogramming, tissue engineering for in vitro systems, and genome editing. Genome editing allows you to actually correct mutations or to generate mutations in your cells, in your pluripotent stem cells, and you can actually develop neurons that contain those mutations. You can engineer the tissue to grow in certain environments, and you have all sort of media and things like that. You can sequence many of the things, because obtaining the pluripotent stem cells, induced pluripotent stem cells, is not an easy procedure. Sometimes mutations happen during the way, so you have to sequence many of the things and make sure that you did the right thing. And of course, the cell reprogramming part. And in the engineering sides, there is in material engineering that allows to have like polymers, like the ones that I was telling you, or bio materials that allow you to grow cells on those biomaterials. Some of them are conductive, and then you can even record from the cells. And the microelectronics allow you to grow those cells actually in place that contain thousands of electrodes. And therefore, probably you have heard about the brain on a chip or BOZ models, which is growing neurons on top of electrodes in a microchip. And in there, you can look at their development. You can look at how they fire action potential, how they network with each other. You can add different things. You can try rescue interventions, and I'm going to show you some examples of that today. This is literally one of the models that these are organoids in a little microplate that we are studying in the lab. So this is one example of that. Now, how can we study? How can we use these model systems to study the link between genetic codes and neural codes? And neural codes are important. There are many people out there studying the link between genes and behavior. But the development of proteins and the development of molecules, it doesn't really match the scales of behaviors. Now I am talking to you, and it is impossible that I'm going to synthesize the protein when I say the word yes or no. What? So behavior is generated by the coordinated electrical activity of neurons in the brain. So there is a neural code that put together these highly organized systems, gene expression, molecules, and the behavior. So you cannot go from the molecules to the behaviors directly. You have to go through the neural code and the electrical activity in the brain, and that is what generates actually behavior. I would like to say that for interaction of thousands of genes in complex molecular pathways, neurons are born. That's how neurons are born from stem cells. In single neurons, such numerous interactions result in the ability to generate fast information carrying events of an action potentials and receptor potentials. And I hope that many of you took the neuroscience courses and you know exactly what an action potential is. Now, when you have neurons firing an action potential and wiring together, there is emerging properties like feelings, behavior, consciousness. I mean, you mentioned that appears there and generates system functions. And those system functions is what the system neuroscience study. I call this like the hourglass of neuroscience, in which on top of the hourglass, you have the molecular neuroscientists. Here you have the system neuroscientists. Everyone is studying behavior, but we don't talk to each other. And there is this narrow stretch here where molecules are becoming electrical events for generating behavior that is hard to study. And I think that the model system that we are looking at is really good for studying that. So I'm not going to talk about a lot of molecular developments today. I'm not going to talk about behaving animals today. I'm going to talk about this narrow thing here that we can study with the iPSC's induced pluripotent stem cells models. So just to remind you, the action, if you bring an electrode close to a neuron, the neuron is firing these action potentials due to the sodium and voltage and potassium voltage sensitive channels. And action potentials or spikes are to the system neuroscientists, which is basically was my PhD on, like genes to the geneticists. They're the events. They're the events that form the neural code, right? And for the neurometric analysis, for analyzing action potentials, we convert it into digital events. It's almost like a computer works out, right? So, and sometimes we convert them back into continuous signals to do some kind of analysis. But the whole idea that these action potentials that the neuron fired is a neural code composed almost of zeros and one, that it transmitted from one neuron to another in a network. And from this activity, there is information encoded in the overall fire rates. I want to show you something really interesting. I don't know if you have seen that. This is one of the videos of the extracellular single cell recording from Hubel and Wiesel and at Harvard University in the 1960s. And they were recording from the brain, the visual cortex of a cat. And I hope that you hear what is happening. You see, right here. Right here is the spot that is the receptive field of the neuron. When the spot of light is in the receptive field of the neuron, and there are other experiments that have been done, that is when we see the spot of light, it correlates with the neural fire. The neurons start firing before we perceive the spot of light. And that's how perception is. That's how high order behavior, like the coordinated activity of many neurons, brain. And the good thing about these events is that we can study them at the level of with the model that we're using, which is the iPSC cells. So, and the idea here is simple. The idea is we take pluripotent stem cells that contain certain mutations. For example, here is a control cell line. Here is a mutation of a SHANK2. This is actually a mutation in a patient. It's a nonsense mutation that I'm going to talk about later. And this is a Rett syndrome mutation. I'm going to talk about these two here. I'm not going to talk about Rett's because it's another story and it would be going too long. But what you can do is you put them on top of a microelectrode plate, and these plates contain thousands of electrodes. In this specific case, we're more dozens of electrodes with this system. And then when you culture the cells will start firing. And what you see here in each row is the firing rate of a cell, basically the action potentials that the cell is firing as a function of time. So this is time, and as you evolve, different days evolve, you can see that there are more cells that they come to fire and more cells incorporate into this. And at some point, you have a full plate of cells firing. And now you have literally a network on a dish. And this network is made of human neurons that are making human synapses. And these human synapses is something that we're very interested in study. And many of you know that in neurodevelopmental disorders, there is a lot of how excitatory inhibitory balance gets altered in those disorders by synaptic proteins and different genes that encode different synaptic proteins. Now, we can apply all sort of methods to study this and to create neurodevelopmental curves. And actually, we could do to one of these networks the same things that Piaget did to the children development, looking at the developmental trajectory of them. But in this case, we have a variable that is, for example, number of active neurons. And I will show you a couple of other variables. Now, I'm going to give you an example of one of our control. This is the normal subjects. And in each row here, you have a neuron firing during a time of 300 seconds here. And here they are the different channels in the MEA. So each row is a neuron. It says channel, but it's basically a neuron firing. And we record during week 1. And record within week 2. Well, there are more and more and more appearing. More neurons are becoming active. So this is probably when they start differentiating from progenitor cells into neurons with all the ion channels and all the capacity to generate action potentials. And now you have in week 3 that there is a very interesting event that start appearing in week 3. Instead of firing all over the place, the action potential start getting grouped into something that we call a network burst. And this is a super interesting event that we're going to talk about. Basically, what is happening is in this week 1, these brown progenitor cells become neurons in gray, and they start appearing. And here they start wiring. And here in week 3, they start actually, they wire even more. So that's the whole idea here. So we have published a few papers on this. And now these events that the condensation of action potential, I'm showing here only one neuron, one channel, is what we call it the network burst. And the network burst is when you see these action potentials that they happen very, very close together. It's an example of temporal summation of action potentials in the, what it does is basically condense the action potentials in a train that is called a burst to produce temporal summation at the level of the synapse. And this is something that we see in these networks. And you probably see in the neonate brain too, when you see these bursts of activity and silent and bursts. And what we think that is happening here is that the brains are wiring and transmitting information. Now, I'm going to talk to you about one of the proteins that is related to autism and some of the neuromental disorders, which is SHANK2. The SHANK proteins are a family of scaffolding proteins that played a crucial role in the formation of synapses in the brain. And I'm going to connect this. I'm going to go back to the recordings of action potentials. Okay, I'm just going to talk a little bit to you about SHANK proteins. So there are 3 main proteins, SHANK1, 2, and 3. And they have a domain where you have, they are located in chromosome 11, and they have these 6 different domains. And these 6 different domains, what basically they do, they anchor different proteins here in the postsynaptic density, so where the neurotransmitter arrives to the postsynaptic neuron. And here you have, for example, anchoring of different types of receptors, most of them glutamatergic receptors, AMPA and MDA. And here you have anchoring of this mGlut-R, which is metabotropic glutamatergic receptors. That is what we're going to be talking about. That now, mutations in the in these proteins, in the SHANK proteins, they are very related to developmental disorders. They are critical for learning and memory because these receptors are critical for learning and memory. There is a very good review of the literature here that you can take a look at. And they have been linked to ASD, intellectual disability, and schizophrenia. In fact, in this review, they talk about rodent that they have the same protein, the shank proteins, and there is these shankopathies, that's what they call it. And these mouse models, they show, for example, repetitive behaviors, problems with brain plasticity and learning. And this is a series of things that you can see in these models. For example, these are mini inhibitory postsynaptic potential. Parvalbumin expression is a protein that is expressed in inhibitory interneuron, and that produces disrupt excitation inhibition balance in these mouse models. There is all sort of things that I wanted to, I probably won't go in detail, just a matter of time. But the whole idea is that these mouse models show defects or deficits that they seem similar to the ones seen in patients. Now, what is the effect of SHANK2 mutations on the function and development of synapses in the networks that I was talking about? So basically, we did this experiment, and that was done in James Ellis' lab. He's the stem cell person. He's really very knowledgeable in that. And this is a patient line, a patient that had a mutation. There was a nonsense mutation. What it does, introduce this stop codon. And the codon, what it does is it produces a truncated protein that is not functional. So the SHANK2 is not functional. And they published a paper on that in 2019. But you can do you can use also a cell line from a patient. From a normal individual, normotypical individual. And what you can do is also to CRISPR-Cas to correct this mutation and produce a control line from this patient line. And that's what they call an isogenic pair. And here we have the control. Actually, you could use CRISPR-Cas also and induce a mutation, which is a knockout, which is basically you get rid of the gene. There is no production of the protein anymore. And then you can take all these 4 different lines, type of cells. So this one contains a mutation. This one contains a mutation, the green and the blue. And these 2 are control lines, what we call isogenic pairs. And you can develop neurons using this NGN2 protocol. You develop neurons, excitatory cortical neurons, and you put it on a plate, and these neurons start firing action potentials. And the first things that I wanted to show you is that, for example, this is the line, this is the normal typical line. And what you see here is that in this case, again, in the row are the different self-firing action potentials. You see that they fire very nice network bursts. So all the neurons are synchronizing here. But when you look at the patient line, so the neurons are going really, really fast synchronization. It's almost like a hypersynchrony what is happening to those cells. They increase the frequency of the network burst, and they are very, very, very fast. So the same thing happens when you look at the control line and the knockout line. So basically in the knockout line, you have a very similar phenotype as the patient. And here you have in this control line a very similar phenotype as the corrected control here. So the yellow is the corrected patient line. So what you will see is that the cells, they're just firing faster. The network becomes, everything goes into faster time scale, and they start firing much. And we call it the hyperconnected phenotype. These neurons are hyperconnected. So the brains of these patients also hyperconnected? This is a good question. But what I can tell you is that definitely these neurons are hyperconnected. When we look at quantitatively at that, and we look at the mean firing rate, so in green here, we have the patient line, and in yellow, we have the normotypical line. We see that these neurons are not only hyperconnected, but they're also firing more spikes. So the neurons are hyperactive. Those SHANK2 neurons seems to grow more dendrites, seems to grow more connections, and seems to be hyperconnected. And the network burst frequency becomes higher. I mean, you saw in the previous slide, but this is basically the confirmation of that. And the mean firing rate, the number of spikes, is the same thing for the control line and for the knockout line. You can see always the blue on top of the pink here. So these neurons are becoming more active, are firing more spikes, and they are becoming more synchronic. And to do that, we had to do a different type of analysis. So we took every single neuron and we computed a correlation between the different channels. This is called functional connectivity analysis. And it gives you an idea. If this neuron is firing, the other one is firing too. So these neurons are not only hyperactive, but they're also hyperfunctionally connected. So the SHANK2 neurons seems to be hyperfunctionally connected. By the way, what I'm showing here is the control. The SHANK2 is this one. So the SHANK2 is the green line. And as you can see, the deficit of the protein, which is a little bit counterintuitive, what it does is produce hyperconnected network. So there is too much connectivity in those things. Now, we tried to rescue this by using a little bit of heuristic on that. So there were some documented studies in mice that were really influential for using this type of rescue compound that was done by Fraser in James' lab, and they had a very good reason. They used this DHPG, which is a potent agonist of group 1 metabotropic glutamate receptors. The glutamate receptors, the metabotropic, they're not ion channels. They influence the ion channels by other means, right? So they are metabotropic receptors. And what they did was to apply from week 1 to week 7, very early. It has to be a really chronic treatment with this agonist of the metabotropic glutamate receptors. And by doing that, the phenotype was corrected. So this align, this correction looks more like this than the I'm sorry, they corrected it is slowed down when this the DHPG was applied. You see that there is a very narrow, very repetitive burst here. The burst here became a little bit longer, and they get slowed down, which is what the normal phenotype has, right? So there was a correction of the phenotype. So in principle, we could rescue those networks. This is just to give you an example of a quantitative example for the correction. Here is the network burst frequency. As you can see, this is the mutant in green, in light green, and is increasing as the number of weeks increase. And the correction with the HPG actually made it slower. So they slowed down. The networks are slowed down with the DHPG. And if you do that in the control, the results is different. So basically the same thing for network burst duration, which is another parameter in here. So we could rescue the networks with the HPG, at least the whole phenotype. So the whole idea here is a little counterintuitive because there is a deficit in the protein, and now the networks, they go too fast, they connect too much, and now we have this correction with the HPG, which is a metabotropic glutamate receptor agonist. So the idea was that these changes could happen for at least 4 things, the 4 C's. It could be a cause, the gene mutation, what I mean, the SHANK2 mutation could be a cause, could be a consequence, or the changes that we see with the gene mutation could be causal, could be consequential, could be a consequence, could be a coincidence, but it could also be compensatory. We had good reasons to believe that these changes that we see in the network, in the SHANK2, with the acceleration of the networks and the increase in the connectivity, are more compensatory than related to these 3 other reasons. So, and there is a good reason for that, because if you look at in the mouse, the data are a little bit contradictory. Because, for example, there is in the mouse, there is this reported spine density, lower spine density and length in the striatum. And what we saw in these cells is the opposite in the human cells, right? So the data are all over the place. So it also depends on the stage of development where you capture the network. That was something that we got from mouse studies. So one of the things that we thought, well, development is a dynamic process, and one must map the developmental trajectory of a system with enough spatiotemporal resolution to detect changes in the system structure and function. So our hypothesis, remember, was that the changes are compensatory, that really what SHANK2 is the deficit. And at the beginning, the synapses doesn't develop very well. And then at some point, the thing compensates and go overboard. So that was the whole idea. So what we did was to use a different system for recording that is called the Maxwell system that has about 26,000 electrodes. In this case, we were recording for around 1,000 electrodes, and they are really very, very condensed, the electrodes, so that we can record from many, many of them, and we decided to record every day. The changes that I explained to you before, we did weekly recording starting at week 4, week 5. So, but in this case, no. We're going to start at day 10. As soon as these cells are put in the plate, we're going to start recording, and we're going to record daily from then, and we are going to see what happens. And this is an example, for example, of a network that we recorded. And can you see that there is more rows coming up and coming up? That means that there are more neurons becoming active and more neurons synchronizing the burst that you can see. As you can see the days here from induction. And it was exactly what we saw in the other recordings. But here we have like a daily shot of what is happening to these networks. And by doing this, we can really characterize the network with a much better resolution. And we may be able to detect if the changes that we're seeing were compensatory or not. And one thing that we observed in those networks was that this is a layout of the electrode plate. And what you see in white is when the cells start becoming active, right? And it's repeated. Top and bottom is the same thing. But at the bottom now, you're going to start seeing red. And in those red, you have channels that start synchronizing. And one thing you may not notice here very well, but I'm going to show on another slide and make it clear, it was that the control networks, they really start with more active electrodes than the SHANK2 network. So in reality, the SHANK2 network apparently was behind at the beginning. So, and this is exactly what you can see in day 13. For example, the control network has a few electrodes, so does the SHANK2. But in day 17, the control network in white has more active neurons than the SHANK2 network. So the SHANK2 network is really behind. It's not an accelerating connectivity, an accelerated development. It's really behind. So, and in day 33, however, this network is all white here. So they have more active electrodes than the control network, the SHANK2. Something is really happening there that we could capture with our high-resolution mapping. And this is exactly what happened. You look at day 17, the control network have many more active electrodes here, many more active neurons than the SHANK2. But if you go to day 33, this is inverted. There's many more spikes here than there. So to quantify this phenomenon, what we did was to plot the number of active channels as a function of days from induction. And this is for the control network. This is day 13, 17, and day 33. As you can see, the number of active channels start increasing and then sort of reaching an asymptotic performance that we can fit it with a sigmoid function. And we can determine 3 parameters of the function here. For example, the time where the network is half developed, which is day 17 here. We could also determine how fast the network is growing, is developing, right? Which is the slope of this function. And what is the asymptotic? How many neurons the network just acquired and stays with how many active neurons. And these parameters allows us to produce a quantitative evaluation of the development of the network. Similar to when you're measuring a child, for example, and you look at the body weight and you do the developmental curves in pediatrics. So interestingly, when we plot the SHANK2 networks, the SHANK2 networks indeed were behind at the beginning. You see this gap in here. I have to tell you that this is still ongoing work. This is an example network that we look at, and we have a couple of other networks that behave exactly like that, or very similar to that. But what is happening is that the SHANK2 is really behind in development. But after day 29 or 28, maybe 27, it overshoots the normal network. And here is where we catch it in our previous work. So we got this hyper-connected, hyper-functional network But what this seems to be happening is that it was behind, and now it just went overboard. And we have some ideas of why is that happening that I could talk to you if you're interested in that. What about network burst? Network burst was the idea of how it gives you an idea how connected the network is, right? How many members of the network are participating in this ensemble, in this wiring frenzy, if you want to think about it in that way. And I told you what about the burst was, this condensation of action potentials here in time. And to make the long story short, when we plot actually for the control network, the number of active channels, you saw this in the previous slide. There is more neurons and more neurons becoming active and goes this way. But in terms of number of bursts, around day 24, the network burst appears in this network, and they stay almost the same all the way to the end. So this is the number of the proportion of bursts that we have in the network. Now, the most interesting thing here is going to be what happened to the SHANK2 network. Do network bursts appear before, after, right? Do they go overboard too, like in the number of neurons? And indeed, what happens to the SHANK2 network was that the network bursts appear after, almost in day 27, but they went overboard again. They have more network bursts than that. So that's the hyperconnectivity phenotype that we isolated. But what we didn't get before was this high resolution mapping in which you can detect that those networks were really behind. And then they actually became hyperconnected. It's very hard to tie this to the clinical side and to the developmental side, but this is at least what we have at the level of the synapses. Now, as a summary, the human stem cells-derived networks and organoids develop following similar trajectories as brain networks during embryonic development. The high-density MEA allows a detailed functional characterization of network development. So synapse formation and the effect of gene mutations, in this case was SHANK2. And these SHANK2 networks show hyperconnective phenotype that can be rescued using chronic treatment with the mGlu agonist DHPG. And the interesting thing here is that this treatment has to be chronic. The treatment has to be start from the beginning, actually from week 1. That's how the treatment, Fraser did the treatment. So from day 1 going all the way, that and this chronic treatment rescue the network phenotypes. So probably producing a more normal development in the mGlu, the glutamate receptors with this pharmacological intervention. And the SHANK2 hyperconnectivity phenotype appears to be a compensatory mechanism to early synaptic deficits, likely caused by the low SHANK2 expression. So not everything, when you do, same thing I guess that happens with patients, brain lesions, developmental disorders, anything, when you capture them, you capture at a moment in time where many things happen. And if we don't have the possibility of map the story, right, of how things happen, it's very hard to establish the cause of something like that. So this is a barcode for Fraser's papers, I believe. So of course, this work was done by Mary in our lab, Kartik, that these are 2 grad students in our lab. Milad is not here. He was a postdoc in the lab that also did some machine learning in this data. And the University of Toronto, in this case, at Sick Kids, James Ellis is the PI with Rebecca, Fraser, Wei, she's just the magician, her and Peter of this stem cell work. They grow those networks amazingly. And at the Salk, I also thank Rusty and Sarah. Sarah is a grad student, and Rusty taught me a lot about stem cells and organoids at the Salk. So thank you very much, and I'm going to stop here because I think I'm a little bit over time.
Julio, that was fantastic. I mean, that's a great review and summary, and really exciting to hear about the future and what it holds. Just you worked with Fred Gage stuff when you were in San Diego. Is that where it was?
Yes, I was in San Diego.
Yeah, for a year. So yeah, it's really exciting.
Yeah.
Because he's a big memory guy, right?
Yeah, he does a lot of things. I mean, he does neurogenesis. and he's doing organoid, Alzheimer's. So the lab, he has like probably 25, 30 people in the lab doing different things, but he's very interested in these fundamental questions about how these models could help to answer fundamental questions for the same thing that I was telling you, because it's so complex. When you get the patient, it's so complex.
Yep.
Everything that happened is so complex. And what I really said was, can I just go to a very simple model and start looking at excitatory synapses, effect of this, for example, nonsense mutation, non-functional SHANK2 protein on that, and can we start from there? And from there, maybe we build the model, we add inhibitory neurons, maybe we add more, we go to organoids, and then we start growing it in a way that maybe we may be able to connect it to the developmental disorders. But in principle now, I mean, you could see that using pharmacology, that was bread and butter pharmacology, we could rescue actually.
But I think it's a really exciting way to look at things, because a resident was asking me, I think today or yesterday, about how correlated executive functioning and intelligence are, and it's probably really almost impossible to measure one without the other, right? So we can't get pure forms of them in clinical patients, and so I think in the kind of things you're talking about, we may be able to sort of look at pure forms of neuronal stuff that we really can't do in humans, and so it makes it really exciting to be able to understand sort of what's actually doing what.
Yeah.
In a lot of ways. All right, so we have time for questions if people have any, or not if, but the questions you'll have. I will start, Julio. So when you were talking about the SHANK2 mutations and the sort of the models, how, and you're talking about the hyperconnectivity, how there's this theory about autism that people with it have abnormalities of connectivity, right? So underconnectivity in certain regions and perhaps overconnectivity in other regions. How does the SHANK2 model you've talked about, how does that fit in with that?
Yeah, it is a really good question, because there are three different SHANK proteins, and what we're thinking that is happening is that there is a compensatory mechanism that when SHANK2 should kick early, and then SHANK3 proteins come into that. And what we may think that is happening is that SHANK2 doesn't kick early, so SHANK3 comes early, but the regulation of the SHANK3 protein is different. And it's very abundant in the brain. I mean, those proteins are in the different synapses. We still don't know very well in inhibitory and excitatory synapses what it does. The development of hyperconnectivity, in this case, these neurons are more on the cortical side. So that's the only thing that we can talk about that. But once you start playing, for example, if you have a hyperexcitable cortex, right? It is possible that you will have a compensatory mechanism that makes basal ganglia to behave in a different way, because this is just sharing inhibitory connections. So the whole network thing, my guess is that this is exactly what I would predict, that if some regions are functionally hyperconnected, you may have a compensatory mechanism in other regions. And that has probably more to do with phenotype and with regulation of the how the phenotype is actually influencing the gene expression, rather than a developmental program that is triggered independently in each brain region. I don't think that is the case. I think that it really depends on how it is affected and which region is producing the most feedback, how the organism the brain start compensating for different things. I don't think that I gave you a good answer, Rob, but I think that my conclusion is that it's very hard to pinpoint exactly what happens in that. These are excitatory cells, and the cortex, when it comes to behavior and high level function, the cortex probably has the most to say. And I guess that that's where you may find most of the...
But so the other question, do people with SHANK2 mutations, do they have a higher rate of seizures?
Okay, I was looking at Reds, they do.
Yes.
But SHANK2, we haven't pinpointed that. And I think that I was talking to James about that, and we were trying to look into that. My understanding, they don't. But it's very much present in schizophrenia.
But I would wonder, though, because if you get, I think one of this theory about abnormal connectivity in autism, I think fits, and with glutamate and things, is the idea that, that if there's hyperconnectivity, does that lead to abnormal sort of, brain circuits by definition, I guess, but does it lead to overexcitability and therefore epilepsy in patients? And because the question, I think, how do we explain the increased rate of seizures in people with a diagnosis of autism?
Yeah, that's a really, I'm after that question, and but I'm not sure that SHANK2 is the most. So first of all, I don't think that there is enough data on that. I mean, we can look at the patients that this cell line came from. I don't think that it doesn't have epilepsy.
Okay.
This patient doesn't have epilepsy. So I think that there are two cell lines. What is the nonsense mutation, and the other one's a missense mutation. I have to look at James because he has the history of the patients, but he didn't have epilepsy. And I asked him exactly this question because in Rett syndrome, MECP2, we find a very clear connection. And we know that in Rett syndrome, a lot of Rett syndrome have epilepsy.
Yeah, and off the top of my head, I think I've only seen 1 or 2 people with SHANK2 mutation. So I don't know. My sample size would not be large enough to comment on whether they really have a higher rate or not of seizures.
Yeah, we'll have to ask some clinical.
Geneticists, I suppose, would probably be the people to ask. So, are there any questions for Julio? Because I think this is a really exciting field, and it really is sort of the future, I suppose, in terms of understanding brain circuits and how they work, and not just brain regions, but how the interaction of different areas. Julio, where does your research go now?
So what we're doing now is trying to understand the networks better. So and now what we're trying to do is, for example, trying to see if we can do rescues with using the stimulation or using optogenetics. But we have a very interesting phenomenon that we describe, which is that these networks, if you take a network and you have neurons and you connect the neurons and you give them some plasticity, degree of plasticity to form connections according to Hebbian plasticity, one of the things that we're thinking is that they spontaneously start forming spatiotemporal patterns. They start forming exactly the same. It's almost like the network, cell 1 gets activated, cell 2, cell 3, cell 4, and it starts activating sequentially. So it's almost like if the network is building, it's like the network is saying da, da, da. So the network is organizing itself. Our idea here is that they're encoding memories. They're organizing themselves to encode memory patterns, for example, and that's exactly what you have in the brain. Now, if you have too much connectivity, like in SHANK2 mutants, the memories may be unstable. And now you go into the cognitive, for example, disabilities or the intellectual disabilities that these patients have, memory deficits. We think that the SHANK2 mutants, they have too many connections, and they become unstable. When you form a memory in the brain, the memory has to remain stable. If not, it's going to be corrupted, it's going to be erased, it's going to be delayed. And what we see in those neurons, very exciting, is that they form these patterns, spatiotemporal patterns, spontaneously. It's just a property of connected neurons that are plastic. I mean, the brain comes with that thing. It's a hard drive that spontaneously start building things in there. And that's where we are going now. We're trying to study the phenomenon and to see if mutations, for example, Rett syndrome, we know that in Rett syndrome, they really go down. The stability of those things go really down, which it's just a cognitive decline that they have when they're 1 year old, maybe, or decline in other motor, other domains. I think that's what we are doing. And in SHANK2, we believe that this is also another phenotype. We believe that there is a U-shaped function in which the synapses need to have the certain amount of plasticity. It doesn't have to, it can't have too little, it can't have too much. You just have to have a certain amount to make stable memories that they're not corrupted, to be able to learn and store learning in the networks. And I think that that's a really good system to study that, how that will going to translate into the patient. And that would be super interesting. It would be to look into that. But yeah, the thing is that this field, we really would benefit a lot from having patient cell lines.
Yeah, so
patients with a mutation. Yeah.
At some point, we could talk with Janice Lim, who I think sees a lot of people with Rett syndrome. But, as you were talking about the overconnectivity thing, I was just thinking about the stuff that you've done with, I think it was macaque monkeys, but around the and looking at attention and stuff. And I wonder if people have overconnectivity, is that going to mean that there's too much background stimulation or too much background noise, which is going to cause problems with focusing and things like that?
Absolutely. ADHD. I think that you're thinking in that kind of
But does that have to do with overconnectivity and there's just too much going on? So, I mean, in a very sort of non-scientific way, it's just too much stimulation, and so they can't focus on things.
Yeah, I think that has to do because in the brain, it's also important to keep to keep things local.
Yep.
Let's say if you have your focus of attention, you have to keep it local to keep a hill of activity there, or a domain where you have when everything is connected to everything, it's not good. So that's really a thing, because it's going to in network theory they produce unstable attractors. So an attractor is something that you go into a state, and the network get into a state, and a state into that state may be a part of the network. The other part of the network is not. But if everything propagates everywhere, I mean, it's not a good thing. You could think about epilepsy. Many people say, "Oh,
exactly."
Yeah. But it's definitely, I would say, that's one of the theories in ADHD, right?
But just think about it with the overconnectivity and stuff. I mean, it just fits so nicely. Maybe that's really, really cool, actually. I mean, that's really, I guess cool is the best word I can use. Just thinking about where this takes us in the future, and really understanding almost at a neuronal level what's actually going on in the brain. Because even, I've thought about this, even when when you're thinking about something and trying to recall something, it takes a little while, then you get it. Like, what actually is going on in the brain? As like, what happens when you're trying to recall a memory? And how does that actually work? It's just, I think, a fascinating thing to even just think about what the process is.
Yeah. I think that there is these networks, they have a property that is called recurrency.
Yep.
Which the activity keeps ruminating there. It keeps like going. And so these recurrent networks that are hyper-connected, they produce, I mean, you can think also about OCD, like that kind of thing that you cannot get off the attractor, and you have something that kicks you out of the attractor. Those networks, they have this property too. It's what I was saying. It is too connected or too little. It doesn't. It's pretty much like almost like the dopamine U shape, but when you take it to connectivity, to the
right, so I guess the balance or something like that, right?
That's right.
Julio, this was a fantastic talk, and thank you very much for doing it. I really, I can only speak for myself, but probably for everybody else also. This was just incredibly informative and educational. So thank you. I really appreciate that. So next month our rounds are on, I believe, May. Actually, I have no idea, to be honest with you. The second Wednesday of May, and I'm going to talk about autism spectrum and how we define it. And we also have our research day on May 29th, and registration is open. And Julio, you and I and Jason are going to meet tomorrow morning to talk about that.
Absolutely.
All right. Well, thank you, everybody, for joining us. And again, Julio, thank you. This was phenomenal. I really appreciate it a lot.
Thank you.
Take care, everybody. Bye.
Bye.